again, it's all, always very difficult to actually follow Jim and Jonathan, because I just want to follow up on some of the things that they've said. It's all very interesting, but I also have a lot of things that I want to cover based on what has been going on, and I'm going to try to finish up the origin of life. But as a result of talking to a number of students here, I have changed this lecture considerably to talk about some early life aspects also, and how that might actually influence an atmosphere. And in keeping with my traditions, this is the Yeti crab that was also first found as a result of the census in marine environments. It's not a very abundant crab, but we've never seen anything like it before. And I'll show you a better picture of it, of the Yeti crab, and why it is very different. And what's interesting is all those filamentous things on their legs are bacteria. It's just wonderful stuff. And so it's just kind of a wonderful habitat. And that, again, is one of the many new species that have been found in the last couple of years. So to continue on in the origin of life, I'd like to follow up on some of the things I discussed on minerals, what I call mineral needs, and then move on to the conditions on Earth that might provide these essential starting chemical energy sources, etc., and then talk about some early metabolisms and what I call the Earth's enrichment period as a result of these new metabolisms coming in. And then, more than likely tomorrow, I'm going to talk about the first microbial community and a possible model system at Lost City, on how we can perhaps view Lost City as a model early habitat for life. So how do we get high concentrations of useful organic compounds? We showed that there's a lot of ways to make them, but if we're looking at this sparsity issue and we want to get lots of the key ones, like the 20 amino acids or the bases needed for nucleic acids, then we want to have specific synthesis. And those are going to require catalysts. So we want catalysts that make a lot of fewer organic compounds. And there's very little data at the present time, but some preliminary data looks pretty interesting. So how do we get these? Keep in mind that the catalytic reactions carried out by proteins in present-day organisms were very likely carried out by minerals before the genetic code and ribosomes. And there's very strong evidence for that, and that's pretty widely accepted by many scientists. Because of the metal-sulfur cluster proteins that are the base of the proteins, like I mentioned, that carry out a lot of the activity. And many of the enzymes, actually, the catalytic activity is at the metal-sulfur base. So what can we learn from that is really important. So some early ideas about minerals and the origin of life. This is not a new concept. In fact, one of the first ones was Karen Smith, who came out in the 1960s with a book that he basically said that the first life was really clay crystals. And for those of you interested in the tradition of this idea, this goes back to the first book of the Kabbalah in the Hebrew religion, where they made a golem, which was a stone figure that can actually do some things. And a number of novels have been written off of the golem concept, including one by a Dutch author called The Procedure, science fiction, where he finds a lot of relationships between the first book of the Kabbalah and how to make a golem with molecular biology. It's a really, really interesting parallel, and so he does that, and he makes life. Anyway, so what Karen Smith's concept is that you have clay crystals that grow and make more clay crystals, but in the process, there's changes in the configuration of the structure, and that's what he's calling a mutation in these structures. And we know that clays attract organic material. In fact, the way that we can actually polymerize amino acids to make peptides, or to polymerize nucleic acid bases to make nucleic acids, we can do that on clays with a wet-dry cycle. And so clays, his idea then is that you can form an infinite number of different crystal structures of the clay, and they're three-dimensional structures. Let's say if they take up amino acids or nucleotides, the three-dimensional structures would produce a peptide or a nucleic acid with a very distinct configuration. And you might actually get millions of those. Oh. This has a mind of its own. That you would get millions of those. You can just stay here and hold it in. Yeah, you can get. It's the Italian in me. I can't keep my hands still. You know? So you can get millions of those, and he basically has what he calls carbon takeover. So that was his concept. Karen Smith is still very active today, and he has some very interesting new papers that have come out that build on this idea now that we know that there are catalytic surfaces on clays. One of the papers that had the biggest influence in the origin of life in the past 20 years, and one that has really spurred on the metabolism first, came from Günter Vechterhäuser and his early papers in 1988 and later papers. Günter Vechterhäuser is a chemist. He's not an academic. He doesn't do research. He doesn't practice science. He's a patent officer, like Einstein was. And so he likes to bring that up all the time when he comes up. Yeah. Have any of you ever met Günter Vechterhäuser? You've met him, so you know something about him. Yeah, he's an interesting person. Anyway,
he's pretty self-confident.
You are really kind. You know, I always like to speak of egos the size of Jupiter, and I've certainly known a couple of those, and Günter is one of those, yes. We can actually name a solar system after him, the Vechterhäuser system. And he would be very happy. Maybe it would be I shouldn't say that, yeah. But his work is really amazingly profound in its influence overall. And so what he said, first of all, he built on the idea of the hydrothermal model that had come out before this in the 1980s. And he said, okay, what is a canonical feature of hydrothermal systems? And that's pyrite, iron sulfides. And pyrites, fool's gold, as Jim talked about early, that's a picture of it. And what he was able to do theoretically was make a bunch of compounds, starting with CO2. I put some phosphate on there, making organophosphates, etc. And you can see in B certain reactions that he has. We'll say from CO2 hydrogen to formate at plus 32 and delta G, which is a Gibbs-free energy, doesn't work because you have to be in the minus level of delta G to make it work. But if you couple the reaction with a reaction of pyrite hydrogen sulfide forming hydrogen, and you couple those two reactions with CO2 reduction, then it's minus 11, which is energetically a feasible reaction to run abiotically and a mechanism that could actually make an organic compound like formate. And what he actually did with this is his first paper in 1988 is 150 pages long. I mean, I don't think 10 people ever could read that paper. It is the most incredible physical organic chemistry paper I've ever read. And so myself and several others said, you know, Günter, what you want to do is get a small paper out of it. Get something that can be readable. And so he took pieces of it and wrote some short papers, but it's had a really big influence in the field. So what Günter was talking about is taking pyrite, getting organic synthesis, and that you would also have condensation, again, surface metabolites, and eventually form information macromolecules in cells. But pyrite was the driver. That was the first mineral to really drive things. In a sense, the Russell model, and Russell has now been, that's all he does is work on origin of life stuff, and with Bill Martin and others, His early work built a lot in some ways on Wechterhäuser, and also pointed out that you can have an iron-sulfur membrane, which can be formed from bubbles. They form as bubbles as a mixture of acidic seawater and alkaline hydrothermal fluid. At the time he wrote this, we didn't know that Lost City existed. So he was hypothesizing that there would be some alkaline system to create that gradient, to make an iron-sulfur membrane. And in that iron-sulfur membrane, then, very based on what Wechterhäuser was doing, except he was enclosing Wechterhäuser chemistry inside of an iron membrane, and having the iron membrane do some of the catalysis. And again, you can see surface metabolites for Wechterhäuser, metabolic pathway for Russell. And from the metabolic reaction came condensation reactions to make large molecules and eventually information. So both have a parallel. Now, I should point out that before I leave this, the group at Carnegie Geophysical Institute, particularly George Cody, but some others, have taken the Wechterhäuser model and actually run experiments on it. And so far, formate and acetate can be made off of pyrite, so that you can make those organic compounds. Some of the ongoing work with minerals, and I'll talk a little bit about this if you're interested. Bob Hazen has a very long article in American Mineralogist that discusses not only the range of minerals, but when they were formed during Earth history, etc. But it lacks in a lot of the catalytic activity that they can do because it's not known. George Cody has spent a little bit more time looking at transition metal sulfides and their catalytic activity. So not just iron sulfides, but other metal sulfides. So if you're interested, these are two very, very good reviews. So what we know about minerals, and I've listed some of them, the main source of phosphate that's going to be around, I think, in the early Archean is going to be tied up in a mineral called apatite. It's mostly a P2O5 mineral that's embedded in basalt. And there's a real concern as to whether or not we can get free phosphate somewhere and how much free phosphate would have been available to make nucleic acids, for example, and high energy bond ATPs. The clays are we've already talked about those. They can organize organics into films. They can actually catalyze polymerization reactions, taking individual amino acids and making proteins, for example. A lot of people are working on clays and condensation reactions right now. And I mentioned pyrite because of its source of reducing power, its ability to reduce oxidized forms of carbon and others. Now, calcite is really interested, and that's one of the minerals that Bob Hazen has been working on quite a bit at Carnegie. And he actually has a paper showing that calcite actually has a chiral preference when amino acids bind to it. So it has a slight preference for L-amino acids when amino acids are bound to calcite. And he thinks that slight, potentially that slight increase in L's in abiotic synthesis would have favored heavier L to D peptides, for example, early on, and eventually potentially L. So that's still an ongoing topic that might be of interest, I think, for others, and something that we probably should think about is what kinds of minerals that actually bind amino acids or bind sugars may have a chiral preference. And I think, Bob is one of the few people actually doing this. The ultramafic minerals are what I'm going to be talking about more tomorrow because they generate hydrogen organic compounds from CO2 and do a lot of wonderful things, and I'm going to spend a lot of time. And I mentioned the borate minerals that can help catalyze the synthesis of ribose sugar. And prior to this particular synthesis in 2004, we thought it was hopeless as to how to make the 5 carbon sugars needed for the backbone of RNA and DNA. So this is just a few of these, and there's a fairly rich literature on the work that's being done with these minerals. So the mineral needs, again, the catalytic metabolic networks that involve the reduction of CO2 to organic compounds, we think is this series of minerals that were necessary. So actually this proto-metabolism may have actually been what I sometimes frequently call the 10-kilometer cell. Not something that's encapsulated in something different, but a wide spectra of area, spatial area on Earth that gives you a flow-through system that gives you a series of mineral catalysis. So think about the early sort of proto-cell as a 10-kilometer entity, for example. But we still need to take all the groups of enzymes that we know of that do certain catalytic activity and try to understand what kind of minerals can carry out those key reactions. We haven't done that yet. One of the things we can do is look at the minerals, or look at the proteins that contain the metals that are present in different minerals and really look at those minerals. We really haven't done that yet. So are there realistic earlier settings that support the metabolism first and the replicator first? And really one of the keys for me is what about the source side of these catalytic minerals? And the answer is we really don't know completely about this. So conditions of the Earth that provide, I think, the essentials for life to start, and I'm going to put some hypotheses in here, that Earth-like planets with active tectonism will support life that uses both chemical and light energy, which I call the unity of metabolism. I'm going to build on that. And prior to the appearance of organisms that actually eat little organisms like heterotrophic eukaryotes, Earth microbial ecosystems would have been characterized by extremely high densities of specific metabolic groups of microorganisms, what I call the enrichment period, a red, purple, or green planet, before you actually had things eating them. This is a beginning of a slide that is something that I got from talking to graduate students here, so this was not something I had planned to talk about. So the enrichment periods would have the potential to affect the chemistry of the atmosphere, and that's what I'm going to try to segue into. So why is tectonism important for life? As Jim said, water is a lubricant. You need water. It generates new crust and continents. It produces hydrothermal systems. And hydrothermal systems are the sources of carbon, chemical energy, and the wide range of 30 elements required for life, from magma degassing, water rock reactions, and mineral catalysis, all the result of plate tectonics. And as I'll point out in tomorrow's lecture, as the possible settings for the earliest microbial ecosystems, and I'll try to make that point. And a possible required setting, not necessarily all of them, but a possible required setting for the origin of life or steps leading to life. And I'll talk a little bit about that. So the unity of metabolism predicts that organisms, even with different biochemistry than Earth organisms, on a tectonically active planet will use the same chemical energy sources and electron acceptors and donors as Earth organisms. It doesn't have to be the same, but if you have a tectonically active planet, you really favor an environment that produces the energy and the oxidants and reductants to keep life going. So the volatiles from magma-hosted and peridotite-hosted vents, and I haven't talked about the difference between those, could they be detected in the atmosphere? And the peridotite-hosted are what we talked about in the serpentinization reaction, ultramafic rock reacting with water. The volatiles from magma-hosted vents are helium-3, which is degassing as a primordial helium, and we use that as a measure to see as to the influence of hydrothermal circulation in the ocean. Hydrogen, methane, and both of those can vary tremendously in concentration. Very high concentrations of CO2, reduced sulfur and carbon monoxide come out of these samples. In a peridotite-hosted system, we have some of the highest concentrations of hydrogen and methane ever measured, formed abiotically, and I'll talk about those tomorrow. But also C1 to C5 hydrocarbons, at least up to C5. That's been what's measured so far, but My French colleagues have just published a paper, or not published yet, that have a spectral data of hydrocarbons where it potentially can go out to C10 to C15 formed abiotically in these systems. We'll maybe talk about that tomorrow. So modeling the atmosphere of a tectonically active planet without life and with ecosystems at various stages in early Earth history is what I think we need to do. That's something that I'd like to see somebody actually do. So,
John, can I make a comment on that? I'm glad you put up those higher hydrocarbons, because Barbara Sherwood Lawler up at the University of Toronto has argued that that's how you tell whether methane is abiogenic or biogenic.
Yeah.
With methanogens, you don't get those higher hydrocarbons.
Yeah, exactly. And in fact, I would make the point, and have made the point at meetings in Mars, that what you would want to do, particularly on something like ethane, the 2-carbon, you still have a very high concentration of ethane. And as you get into higher numbers of carbon, you really decrease the concentration. But you still have a significant amount of ethane and propane. And those are very volatile going to the atmosphere and have also a lifetime in a Mars atmosphere that you could calculate, you know. And so those would be very important to look at. So Earth without oxygen and eukaryotes, so it doesn't have oxygen and it doesn't have animals to eat things. It would be the absence of animal predators. That's the first couple of 100 billion years of Earth history. And so that can result in an enrichment period. And so as you develop a new metabolism for the first time, let's say, I am the first photosynthetic organism and I'm going to use hydrogen and light. There's no one there to have anything to do with me, and so I can go up there and just make a living and produce billions of myself until I run out of something. And it could be, you know, phosphorus, it could be anything, but for that period, I'm going to be full tilt boogie. I mean, I'm just going to take over. And I think, I don't know, for those of you who know Janis Joplin and that era of rock, then you'll know what full tilt boogie is all about. I'm sorry, I can't help being who I am, and my past history comes out periodically. So the enrichment period of Earth history, and so what I call a pale dot of different hues. So rather than the blue planet of Earth, we might actually see a lot of different colors on a planet, on Earth even, at a different period. So this is just another depiction of something very close to what Jim has pointed out, which is in billions of years of Earth history, going back to the formation of the solar system and the formation of Earth. We think that, depending, you know, from what we heard both Jim and Jonathan talk about, the possibility of either liquid water in the subsurface or an ocean as far back as 4.3 billion years or so. And then you have this period of late heavy bombardment. And where we actually do know something about organisms is in that early Archean. The cyanobacteria go back to about 2.7 billion years based on a number of different analyses. Microbes could be out to more than 3.5 or so billion years, the sort of blue arrow. And based on this, we have an ocean that in the Archean that is very high in reduced iron. And you will see later that that goes away primarily because of photosynthesis early on in that period. And we have a sulfitic ocean. This is part of the Canfield model for the ocean. And then eventually we have these early and late freezing cycles that Jim talked about. And then in the Phanerozoic, we developed the complex animal and plant environments. And our oxygen now becomes very high level. And I've plotted Mars on top to get some idea of what we think with an atmosphere loss, primarily at greater than 3 billion years ago. So what happened in the first few 100 million years of Mars would be a very, very interesting thing to model for me. And if it takes a very short time for life to start, I think the water may have formed even earlier on in Mars than on Earth, and perhaps life started on Mars and through panspermia seeded Earth. I mean, we don't know that. Some people have speculated that we're Martians or vice versa. And I'm just showing this. This is an old thing that I made because Jim talked about it. I just wanted to highlight one of Sleep's papers that came out in 1989. This actually followed a Mayer and Stevenson paper that did the same thing earlier on. Just didn't get as much publicity, I guess. But the gray marks are the craters on the moon, and from that was modeled what the impact would actually be on Earth, and taking into consideration also as Jonathan pointed out, the larger size and higher gravitational pull of an Earth versus Mars. And so what was interesting, and the reason I'm showing this is, in this paper back in 1989, Norm Sleep and his co-authors pointed out that you would actually have,
Sorry, John.
did that leave again?
We tried.
That you could have enough of these size bolides to actually evaporate, let's say, a 3-kilometer deep ocean. And one of the reasons I'm pointing this out is because it was Norm in this paper that said that life started in the subsurface. That was the conclusion of this paper, and it really was a very influential paper on me also. And Jim also mentioned the Abramov-Moches paper, saying that heavy bombardment would not have sterilized the Earth. I just happened to have this in here, but also to make the point again that my view is that anything that happened to Earth might have been, particularly early on, might have been beneficial to the origin of life, and I'd like to think about it that way. And as I said, one of the things about a heavy bombardment is it might create a drying wet cycle. And a drying wet cycle on minerals could make polymers. And it may have actually been a very essential component. Just like the late Stephen Jay Gould mentions that the extinction events, and particularly and the extinction of the dinosaurs allowed the mammals to radiate out and eventually become Homo sapiens. Perhaps without those impacts, we may not be here. And how many of you have read his book called Wonderful Life? Yeah, I think it's a wonderful book to read. Have you read that? It's a great book. It's about the Cambrian explosion and how he sees the Cambrian animals in terms of their evolution into mammals and into us. And so he talks a lot about evolution, and he talks a lot about contingency. And that is a chance that, that will fit the rare earth thing very, very well that you're going to talk about on Saturday. It talks a lot about contingency and how rare it is that we actually exist. So to give a little timeline here, so the origin of life is somewhere up here in probably occurring in about a 400 million year time scale. The tree of life indicates that high temperature methanogens and sulfur reducers and nitrogen fixation are very, very old. Also, anoxygenic photosynthesis doesn't produce oxygen is also very old, and there's evidence for this even at older than 3.5. So, let me see. So Earth life requires the elements, volatiles, and minerals produced as a result of tectonics. I've already said that. So how important is that? And I've taken this from one of Don Canfield's paper, and I've modified it to put metabolites in the atmosphere just for this group. So I don't know how, this is something I had to do last night, so I'm not sure how I'll be interested in a discussion of it. But the key electron acceptors, or donors rather, that drove all the early metabolisms, hydrogen sulfide, sulfur, iron, methane, ammonia, and various organic carbons, which he puts in actual sequence of availability, I think, very early on. Note where they're from. Submarine hydrothermal systems, volcanoes, submarine hydrothermal systems, hydrothermal vent. It's these volcanic environments that are producing all of the electron donors that are driving these kind of metabolisms, methanogenesis, photosynthesis, acidogenesis, which is like methanogenesis, except rather than methane, it makes acetate, a 2-carbon acid. Again, in the following anoxygenic photosynthesis, it goes from hydrogen, hydrogen sulfide, sulfur, and iron, and eventually water. So if I had as each of these metabolisms would have emerged, let's say methanogenesis and early anoxygenic photosynthesis using hydrogen, we would expect to see a lot of methane produced, a lot of hydrogen sulfide, nitrous oxide, N2, and perhaps a rundown in CO2. So I don't know, how we would actually, map out these ratios, but it would be really interesting to look at those. And the same with an H2S-driven anoxygenic photosynthesis. All of these could actually have some effect on the atmosphere for short periods. And it would be really interesting to model just this as part of Earth history. And this is a paper modified from Olson in 2006 to add some new information. But we look at the reductants for photosynthesis. There is early evidence from carbon isotope fractionation that I think both Norum and Olson and others think the only way to interpret that is that hydrogen was driving this life form. And they think that this may be the first photosynthetic organism at 3.8 billion years with hydrogen. So this is, I've highlighted in red because I don't know how true that is. The best evidence for hydrogen-driven photosynthesis is from the buck reef microbial mats, and there's some new evidence that Norm and his group published in 2010 on this. And in the Warrawoona evaporites, it looks like hydrogen sulfide might have actually been running the show there. And so you can work your way up. It's primarily hydrogen, and then hydrogen sulfide, in which we see those in barytes, and then iron oxide. We've certainly seen that group of organisms, and then eventually the splitting of water. Now it's interesting that the metabolism of all of these organisms for photosynthesis do not have the same metabolism as the water splitting ones. The water splitting ones all have one kind of metabolism, the Calvin-Benson cycle. All of these have at least 3 different metabolisms. So there were a lot of experiments and metabolisms going on for photosynthesis, and all the early ones involved non-oxygenic photosynthesis with electron donors not being water. It's when these ran out that it favored this particular process. The other thing I'll talk about perhaps later is that this pathway here for reducing carbon dioxide with light and splitting water is a pathway that can completely tolerate oxygen. These pathways don't tolerate oxygen. So it's kind of interesting that the early CO2 fixing pathways that came about had enzymes that were sensitive to oxygen. So when oxygen formed, these guys have to retreat into environments where there's no oxygen, and that's where we find them today. What's interesting about many of the anoxygenic photosynthetic organisms is that they photosynthesize out in the infrared region. They actually have chlorophylls that absorb in the 600 to 800 nanometer range. And so what they have done, I think, early on is avoid ultraviolet light by actually evolving a photosynthetic system that works in the infrared. Because I did think it took some time to actually get up into the upper water column and use visible light range, particularly 420 to 450, which is what most things do. And for these organisms up here, they've developed ways to counteract ultraviolet light, and I mentioned carotenoid pigments and repair mechanisms. I've pointed out a couple of these. The purple sulfur bacteria, for example, which reduce CO2 with hydrogen sulfide, produce sulfate. And we have some, evidence here in the barytes that this is the kind of reaction that was going on. And in fact, maybe the early sulfates entering into the ocean had nothing to do with oxygen, but had to do with an anoxygenic photosynthesis. So we produced this, one of the really most important substitutes for oxygen for organisms to breathe, to actually get their oxidants from, in the absence of oxygen. And so you can make a strong case that early on some of the first sulfate being formed was done not as a result of oxygen. And this is how the iron oxidizing bacteria that photosynthesize work, is they take reduced form of iron, which then can actually reduce carbon dioxide and make organic compounds and iron hydroxyl groups and protons. And all of these we know are fairly ancient. Now this is one I want to point out. This is a very interesting paper that's been followed up a little bit, the Beatty with also Bob Blankenship, who I know. What they found is an obligately photosynthetic bacterial anaerobe, non-oxygenic, in hydrothermal vents living on black smokers. Now, the reason why this experiment was done was because Cindy Van Dover, who's a hydrothermal vent biologist who studies animals, was really interested in this because she found a shrimp at the Mid-Atlantic Ridge that had a kind of an ice spot on it. And why was something living, in this case, at more than 3,000 meters in the dark, have this ice spot? And so she had the idea that maybe these black smoker sulfides were emitting some kind of black body radiation. And so she took down the necessary instruments to the bottom of the sea and did find that, in fact, these black smokers were emitting black body in the 700 to 900 nanometer range. So from that, they said, can there be some kind of photosynthetic bacteria down there? And so this group went down and they isolated a green sulfur photosynthetic bacteria that is capable of this reaction using hydrogen sulfide. And the interesting thing is it's an obligate anaerobe. That is, it's very sensitive to oxygen. It can't grow in oxygen. So there's no way for it to be transported in the ocean and survive. And so their view is that, and Bob Blankenship is actually kind of keen on this, is that perhaps photosynthesis did not occur within the visible light range at all. It occurred early on in the deep sea associated with black body radiation. And so that's when all the evolution occurred. And that's not been proven, but that's a very provocative idea that you can actually have, if you have radio decay of some kind, and it can produce black body radiation on certain structures, that you might actually fuel something resembling. So this is a very provocative and very interesting piece of work that could actually have a profound influence on how we think about planetary photosynthesis. So just keep in mind that nothing is as simple as we think. So this is what an enrichment looks like. This is something you can do in the laboratory really very easily, is just get a big tube and go out into a salt marsh somewhere and get some sediment that's reduced a little bit, that's lacking in oxygen. And what I normally do is I cut up a few newspapers and put them in there to give some cellulose to get microbial activity going on down here. And so first thing that happens is it turns completely anaerobic, and you have sulfate reducers produced that produce hydrogen sulfide. And as the hydrogen sulfide goes up, it stimulates these purple sulfur and green sulfur bacteria that make these wonderful purple colors and things. And you can do this, this can happen at room temperature in a couple of weeks. It's a great demonstration of this kind of thing in a laboratory. I do it all the time. I mean, this is something that, okay guys, this is what we're gonna do today. And and then we sample them usually, so get to see what these organisms actually look like. So these are some of the enrichments that already have occurred on Earth and still occur on Earth. And I showed you these salt beds, the halophile world, of evaporites. If we had long areas of evaporites so that you got high brine, these organisms would dominate. And they have a rhodopsin pigment that comes out very red. And if you fly over San Francisco Bay or other salt flat areas, you'll just see, tens of kilometers of redness. So in anoxygenic photosynthesis, you might actually see long areas on the purple end. Oxygenic photosynthesis without predators, you may actually see something more on the green end. I'm not quite sure what a methanogenic world is. The closest I've ever seen a methanogenic world is after Mount St. Helens blew up, and all the lakes in there had turned black from breakdown of organic material. But it was like a boiling water. It was just bubbles pouring out of there, going out into the atmosphere, and they were all methane bubbles. And that was the most I've ever seen of what a methane world would actually look like when it just goes crazy. An interesting one, a paper that came out in 2005, found a very large area of light coming out of the ocean. So it's this big glowing area that's about over 1,500 kilometers of the Northwest Indian Ocean. And this is the size of some states. And what it was a bloom of a luminescent bacteria, a bacteria that make light. And so it just, the right optimum conditions came up for this bug to make light and made this giant area that can be actually seen from space. So my point is that enrichments like this do occur on Earth today, and we can observe them. I don't know the extent. I'm putting this stuff up for you guys to think about. What are there some different ways to think about what we should be looking for on exosolar planets? So back to the hydrogen world. Today, there's a lot of organisms that use hydrogen. And of course, in these lost city samples, I'm going to talk about hydrogen is a very important component of the energy source. Magma-hosted systems also, hydrogen is a very important in hydrothermal systems. Photosynthesis, etc., a lot of them actually produce hydrogen, and there's hydrogen-driven ecosystems, and particularly in the subsurface. So the evidence points to hydrogen as really the earliest source of chemical energy, both non-photosynthetic and photosynthetic organisms. So hydrogen was key. Hydrothermal vent environments would have provided that hydrogen along with some of the other components needed for life. And evolutionary phylogeny of extant organisms really support the hypotheses that hydrogen-utilizing high-temperature microbes are very ancient. So it's kind of a summary of what we had. And I've shown the Norm Sleep paper, Nesbitt and Sleep paper, where he believes hydrogen was the driver of the first group of organisms. They were high temperature or hyperthermophile, and they made giant mats or biofilms. And this paper is kind of interesting because it follows on the bacterial lineage. He has hydrogen utilizers and anoxygenic photosynthesizers coming fairly early. Sulfur producing and are processing and methane producing organisms also very early on in Earth history. And so what are the lifestyles of hydrogen utilizing anaerobic microbes that we know of? All form complex microcolonies and biofilms. In other words, they like to be together. And that's just, you frequently see, and I'll have to describe a biofilm later on because they're cool. They act like multicellular organisms. So they're no longer a single cell bug. They talk to each other, they do all kinds of things. I want to write a science fiction novel that takes a complex microbial community and create intelligent life from microbes. And I think it's possible. I don't think we need to have us to have intelligent life. The problem is how we can get microbes to do technology. That's been a big problem with me. But I'll work that one out. I'll have a dream. You know how much science has actually, breakthroughs have come from dreams? How many of you know what the benzene ring looks like? It's a 6-carbon ring. It makes interesting things like phenol and toluene. TNT comes from benzene, three nitrates and a methane. Well, this was discovered by an organic chemist in the 19th century, German Kekulé. And the way the story goes is that he was just so frustrated he couldn't figure out the benzene ring structure that apparently he was staring into the fire and drinking a lot. And then as he was drinking a lot and into the fire of this fireplace emerged the structure of the benzene ring. So I've tried that, to see if I can get a better insight in the origin of life, and I just fell asleep. So no insight has come out of it. But you guys should try that. You just never know what might happen. But so they form these colonies, and most use CO2 as a source of carbon, and sulfur or iron as electron acceptors. They grow in most of the extreme anaerobic environments, and those from extreme environments usually grow as these complex biofilms on minerals. So pyrites, calcite, basalt, etc., they all grow on these. The first photosynthetic bacteria probably used hydrogen as electron donor. So there's a lot of very interesting metabolisms and lifestyles of hydrogen utilizers. And I thought I would make a couple of comments. What is the likely metabolic pathways of the ancient? And this is a complicated slide, but I just thought, what the heck? First of all, in this slide, what I want to point out is that this is all the models for where life might have actually started. I've just looked at all the literature, and this is what I've come up from. Intertidal zones, tide pools, sand, freshwater pools, ice fields, etc. You can go down a very interesting paper in radioactive beaches because the possible role of actinides. But the point is, and then each one of these has, why it actually works. So all of these do something. So ice fields can concentrate, organic materials can be concentrated in these. You can make organic compounds. But something is out of order here. So anyway, summarizing the case, this is actually not what I want. Am I moving in the wrong direction? So anyway, I'll start with here. So hydrogen-based ancient metabolisms, again, they're CO2. They occur under anaerobic conditions, and the reactions are catalyzed by minerals, etc. And this is where I wanted to get to. This is the different carbon dioxide fixation pathways. I just want to point out that I have 5 here, and these are unknown. 2 of these now have been decided on in the last year or two as modifications, a little bit of some of these. But the point that I want to make is that the 2 pathways that people are very interested in that include organisms that use hydrogen are this pathway that most of the methane utilizing organisms use, and this pathway, which is our energy ATP producing pathway that we have in the mitochondria, but when you go in the reverse it fixes CO2. And so this is called the reverse or reductive citric acid cycle, which is thought to be also fairly ancient. And these are hydrogen utilizers. This particular pathway is an anoxygenic photosynthetic organism. These pathways contain archaea, high temperature archaea, and photosynthetic organisms. And these unknown pathways are only high temperature anaerobes, hydrogen utilizers. Now what is interesting is that this pathway and these pathways all have enzymes that are oxygen sensitive. This pathway has some, I call it anaerobic or microaerophilic, but they have some enzymes. Now the Calvin-Benson cycle, none of the, which is where all plants, cyanobacteria, and many of the chemosynthetic organisms that use oxygen, this particular carbon dioxide pathway is the only one that's not sensitive to oxygen at all. And so I really make the point that in order to get oxygenic photosynthesis, this pathway had to be evolved. Now what's interesting is this guy, Ignococcus and related groups of organisms like Pyridictium, when they first went in to see what kind of metabolic pathways they had, they couldn't find any of these, but they found genes for a couple of these. So Pyridictium, for example, has one or two of the genes found in the Calvin-Benson cycle, slightly modified. I don't know what they do. And this came out when a methanogen, Methanocaldococcus yanashii, which was the first microorganism to have its complete genome sequenced, they found a gene for rubisco, which is one of the early genes found in photosynthetic organisms. It's the only Calvin-Benson cycle gene in the methanogen, and it actually is expressed. We know it's expressed, but we don't know what it's doing. It's just that there. So the point is that it's probably from these guys and from the Archaea that this pathway actually evolved. And so that's a big area now, is these organisms with what we call chimeric pathways for CO2 fixation. In other words, they have mixtures of these components plus some unique ones. There were a lot of experiments going on how to fix CO2. That's my point. Lots of experiments. And I'm positive that there's still more organisms with unique CO2 fixing pathways than what we have. And eventually, when we get enough of them, we'll be able to understand the origin of this pathway, which I think came much later. But I think this whole concept of how you fix carbon dioxide and when and how evolution moved in that direction was extremely important. These pathways are this pathway is very ancient, probably the first metabolic pathway to exist for a microorganism. So in summary, there are hydrogen-utilizing microbes that use most of the metabolic pathways. Obligate hydrogen-utilizing microbes, including the methanogens, sulfur utilizers, etc., are totally anaerobic and use those anaerobic pathways. All of the pathways are found in hydrothermal vents, including at least two that we don't know anything about. So every CO2 fixing pathway has a representative or more in hydrothermal vents. The point to make and hypotheses here, and which is going to transition a lot more into tomorrow too, is a dynamic ocean subsurface circulation system was necessary to transport various organic compounds synthesized under a specific set of conditions to other environments for further reactions that would result in a greater diversity of organic compounds. One sentence, including organic nitrogen, sulfur, phosphorus, etc. This is when I call the 10-kilometer cell concept. This is what I mean. That if you're going to have metabolism run primarily on this kind of mineral system, those minerals are formed under different sets of conditions, and we need to come up with this dynamic circulation system. And a narrow subset of the conditions up here spawned and maintained early life forms. So what I want to do is decouple the environmental conditions that led to life to the narrow conditions that first. This is totally against the grain of most origin of life people who want to find one environment where everything can take place. I'm saying that we want to use the whole Earth to help us get to life. And that's why I'm even looking at impacts, everything. How can all of this help us get to life? And this is a figure that is going to go into a big paper that me and a bunch of students and postdocs that have been taking my Origin of Life course for 3 years have finally put together. But what we've done is take an overview of all the sites present on the early Earth that may have contributed to the origin of life. And so you see vents, perhaps beaches, and we discuss all of these and talk about the ones that have the most favorable set of conditions for various reactions, and then also point out where there could be greater circulation. Blue arrows indicate some water flow, but we actually have a little bit better circulation model. And what it comes down to, just to go quickly, is that the two environments that are going to be the best for hydrogen-based life are going to be these hydrothermal systems, and I want to make that case eventually. And the main case that I want to make, since I'm running out of time, is that there is a higher probability for life and a separate origin of life on a tectonically active planet with water and volcanism for all the points that I made earlier. The need for hydrogen to run the system, the need to get the key elements and sulfur compounds, etc. And I think, you know, our molecular evidence shows that would be what we'd want to find. And this is where I'm actually going to end. There are two kinds of hydrothermal systems, and I'm going to talk about these in great detail tomorrow. The magma-hosted systems, where there's extensive surface and subsurface environments. And a number of you have asked me about what goes on in the crust, so I'm going to use this little discussion tomorrow to talk about the data that's available in the crust. And the volatiles and elements from water-rock interactions are extremely important here. And peridotite-hosted systems occur mostly off-axis from spreading areas and slow spreading areas. A very different chemistry. And we're finding in the last few years are lots of mud volcanoes in the ocean. In other words, we see these mounds in the ocean. They're mud. But when we sample them, we see that it's buried deep down somewhere, peridotite-hosted systems. And in serpentinization reaction, which I'll talk about later, when olivine or ultramafic rock react with water and get hydrated, there's an increase in volume of that rock by 40% because it's been hydrated. And so what happens when you increase the volume of a rock by 40%? You create a ton of cracking, you do all sorts of wonderful things. And I'll indicate why that is important and what that would do on an exosolar planet if we had that going on. That could be very, very interesting. So I'm going to end it here, and then we'll talk about hydrothermal systems tomorrow, and hopefully I'll be able to get into my own personal environment, the lost city, and try to use that environment to synthesize a lot of what I've been talking about. So let's go have lunch. I have probably time for a question or two if anybody.
We talked about apatite, which is a form of phosphate mineral that doesn't really react very well with organic compounds. Where do you see reduced phosphates being generated in these environments, playing a role in the earliest evolution of life?
Yes, really, that's one of the best and toughest questions to answer. And the phosphate cycle in the really early Archean is just not known. I don't know if you know anything about it, but there's some real phosphate experts that I've met with at meetings, and they don't really know. And I think one of the reasons why the paper came out on arsenic, really, if you want to know the truth of that, is that arsenic is coming out of these hydrothermal systems as a soluble form of arsenic. And we don't see phosphate in that way. And phosphine as a phosphorus gas has been measured, and there's a cycle there, but nobody's really looked at it. So I think one of the things I do, okay, so I'll tell you one of the things I do that I'm not going to talk about, is take a look at apatite, take a look at the mineral that is P2O5, and grow some of these organisms that I think are fairly ancient on medium with only apatite and no source of phosphate, so that they can form biofilms and dissolve that. The problem that I have is that the microbes are incredibly efficient at getting phosphate in very, very low numbers, so my controls haven't worked for me very well. But that's what I'm working on, is that I'm trying, because a lot of these bugs are already known to attach. In fact, I have one that attaches to iron minerals and gets all its iron from minerals, gets its nickel from minerals, gets all the metals it wants from minerals. So I think dissolving solid phase phosphate may have actually been a really important component. And that's why I probably should talk a little bit later on I only have 3 more lectures about biofilms and why they are really important and what they can do that a free floating around microbe can't do. If you're a biofilm on a surface, you can do a lot of things, including get things in the insoluble phase. So Jonathan's question is really important, and I can't tell you how many times I've had a discussion on early phosphorus cycles. We just don't understand.
We showed the absorption of chemicals on pyrite, that they can form bonds that would never have been formed before. Dealing with phosphate, is it also possible that this pyrite enhances the possibility that phosphate to phosphate reaction is established so that you get energy bonds like precursors of ATP already on pyrite?
Good point. I mean, Gunther Vectorhäuser talks about that, but he doesn't really have a model for that. But that's really an interesting factor, that you might actually be able to do that. And